So for 11.12, this one's pretty easy. We're going to summarize and compare the properties of DNA polymerase 1, 2, and 3. Okay, so let's just make our table here. So DNA polymerase 1, polymerase 2, and polymerase 3. So can they initiate synthesis? Can they on their own? No, they can't. What do we mean by that? They need a primer. They need a 3' hydroxyl of a nucleotide to add a nucleotide to. So they cannot initiate synthesis on their own. So that's why we have that enzyme called primase to help with DNA replication. So none of them can initiate synthesis. Can they polymerize in the 5' to 3' direction? And what does that mean? So here's our template down here. And I'm going to put some bases here. And I'll put A, T, G, C, C. Okay, so if this is our primer, the last base of our primer, which is going to be a U. I guess, wait a second. Primase puts down an RNA primer. Yes, so that would be a U. So we have an OH group here. Okay, so they can all polymerize in the 5' to 3' direction. In this direction, 5' to 3', let's call it synthesis. Polymerization or synthesis, whatever you want. You can use either one. So they work in the 5' to 3' direction. And this direction refers to the strand that is being replicated. So this is the strand that's being replicated. So we're going this way, 5' to 3'. The 3' end is going to be over here when we're finished. Okay, so can it do that? Yes. So it's going to put in A, C, G, and so on and so forth. And there it's going from the 5' to the 3' direction. Okay, so they can all do that. Let me move this down here. Move this down right about there. Okay. So they also can all, what else can they do? They all have a 3' to 5' exonuclease activity. So 3' to 5' exo. And that is used for proofreading. I'll put the pluses up there for the 5' to 3' synthesis. And I'm going to put the pluses down here for 3' to 5' exonuclease. Exonuc. So they can all do that. And what does that mean? Well, that's very important that they can do that, because if they should make a mistake, no one's perfect. Genetics professors aren't perfect, and DNA polymerases aren't perfect either. So let's say this is, okay, I was going to put a C, but that would be correct, so I'm going to change that to a G. Let's say it puts a G in here. Well, G isn't supposed to be paired with G. So the polymerase can, instead of going forward, some mechanism, and we'll probably talk about this in molecular biology. I'll be teaching that next year. But we can talk about why it pauses. Somehow it pauses and realizes it made a mistake, and it backs up, goes from the 3' to the 5' direction, backs up, and cuts this out. Exonuclease. So nuclease is just cutting DNA. Exo, you think of that as cutting it out. Exonuclease. And does this in the 3' to 5' direction. So we're going to cut that G out, and then it can move forward again and put the right base in. Okay, so they all have that 3' to 5' exonuclease. Okay, and there's another one that's important. If you remember this, then that will allow you to remember some important things about DNA replication. So 5' to 3' exonuclease. So that's just, cutting bases out that happen to be in front of the polymerase. So only one of them has that activity, DNA polymerase 1. So no for 2, no for 3. And okay, what does that mean exactly? Well, that means that this polymerase is like running down here. And okay, well, you know what? Let's say, well, we'll do RNA bases in blue. Let's say there's an RNA base here from an RNA primer that was put down before in like an Okazaki fragment or something. Well, DNA polymerase 1, if it's polymerizing, what it can do is if it runs and runs into a nucleotide, well, it can cut rid of that. It can get rid of that. It can cut it out. It's going in the 5' to 3' direction, synthesizing, and it bumps into a nucleotide that's in its way. Well, it can cut that out and keep moving in the 5' to 3' direction and put down the DNA bases. So the others aren't going to be able to do that, and they're just going to have to stop replicating. So let me put down the G here. Okay, and it can keep going. Okay, so those are the main properties of DNA polymerase 1, polymerase 2, and polymerase 3 that we will need to remember. Okay.